Hey everybody, Mr. Larson here. So today we're going to be talking about DNA and what it's good for. So before we get started into what DNA is, we have to understand the background of it and the history of figuring out what DNA actually is and what it looks like. So this all started off back in 1928 with Frederick Griffith, where he was trying to figure out how bacteria was making people sick. So what he did in his experiment here above is that he tested a couple different types of bacteria. So the first bacteria, when injected in a mouse, actually killed the mouse, unfortunately. But the second bacteria, which is a completely different one, when injected in the mouse, allowed it to live. So he was kind of intrigued by this. So first he tried to, with the bacteria that normally kills the mouse, he heat tested it, and when he heat tested it and injected the mouse lived. So he was confused. Why would this bacteria that normally kills a mouse, when it's heated, why would it not kill the mouse? So what he did is he took one sample and put it in a different petri dish. He put another sample in the petri dish, allowed it to bake overnight, and then combined them together. When he combined them together and injected the bacteria into the mouse died and actually had that smooth bacteria which caused the disease in the first place. So he was confused of why this was happening. So when he mixed the heat-killing bacteria with the harmless bacteria actually transformed into the disease-causing bacteria. This is a process called transformation, in which one strain of bacteria is allowed to change the genetic material of another piece of bacteria. So this actually even happens in our own bodies. In 1952, another scientist named Rosalind Franklin was able to use X-ray diffraction to create an image of DNA. She was able to figure out that it actually had an X-shaped pattern in the DNA. I'll actually show you the picture here pretty soon. She actually died of cancer at 37, just due to X-ray diffraction. There's so much radiation that she's been exposed to. So that might have actually caused the cancer to grow within her. So this is the image that actually she discovered. You can see here, you have this round circle going around it, and then you have this sort of X that goes through the middle. Because this was way back in the day, they weren't actually able to have that 3D image that we know of today. So this is actually the very first image of DNA ever discovered. So in 1953, Watson and Crick actually found that picture, and they started developing this idea of a double helix model. So they were the first ones to ever come up with this idea where instead of it being a ladder, it's actually sort of a twisted ladder, which we'll see here in a little bit. So DNA, what does it stand for? It stands for deoxyribonucleic acid. This is the molecule that has all our genetic information. If you remember back in the biomolecules, we talked about nucleic acids. So this is one of those nucleic acids. So deoxyribose is one of the other main portions of it. So it's something similar to like fructose, glucose, which we learned about in photosynthesis, which means that this is a type of sugar. So it's a nucleic acid with some form of sugar attached to it that contains all our genetic information for cell development and function. So just like Watson and Crick, their idea was pretty spot on, where it's this very long molecule kind of spiraled together that is made up of different units called nucleotides. Each nucleotide has 3 basic components: a 5-carbon sugar, which we will see here in a little bit, a phosphate group, and a nitrogen base. Here's an image of our nucleotides. So here you can see this little ball here is the phosphate group that kind of bridges that gap together. These are your 5-carbon sugars. This is your deoxyribose right here. And these are your nitrogen bases. Each of them is different structurally. So if you look, actually, they may look different just because they're different colors, but when you actually look at the chemical formula, it actually has a different shape, a different type of structure. So yeah, you have your adenine, your guanine, cytosine, and thymine. These are your 4 types of nitrogen bases, which we'll start to look at here. So just as I mentioned earlier, your adenine, guanine, cytosine, and thymine are all your 4 nitrogen bases. These are your only 4 bases that are ever in any DNA molecule. There's no more, no less than these 4. So every single all your genetic makeup is made up of these 4 bases. So you can also see them as A, G, C, or T, corresponding to their name. So here is the image of the structure of DNA. Here you can see the double helix model. So it's kind of that twisted ladder shape. You see here that C is actually bound to the G. So this is how it always works. C is always bound to G, A is always bound to T, and T is always bound to A, and C is always bound to G, G is bound to C. This never changes in a DNA molecule. So in DNA, these pairs always happen. They are bound together through what are called hydrogen bonds. It's just sort of an attraction of these 2 different types of bases. But this system never changes in any DNA molecule. It's always C to G or G to C, as well as A to T or T to A. So here you can see the sugar phosphate backbone. That's sort of that outside section. And then you see the binding nucleotides in the middle. So base pairing. Hydrogen bonds, just as I talked about before, are the only form of basically binding that is bringing them together. So they only form between specific base pairs. You have your adenine and thymine, just as I mentioned before, A to T, and then guanine and cytosine, so G to C. Those are the only 2 pairs that happen. G will never, ever, I'm going to read it one more time, ever bind to T or A. It is only C is its best friend, and it will only hang out with C. Same thing with T. T will never bind to G or C. A is its best friend and will only hang out with A. So that's basically how they form together. So that is the structure of DNA. If you have any questions, please feel free to ask. And please continue on with the digital lesson. See you soon.